I think we'll like to start the hot topic session now. I'm delighted to welcome Professor Alexandra Worden of GEOMAR, where her research focuses on the regulation of photoautotrophic microbes with an emphasis on carbon cycling in marine systems. Today we are lucky enough to hear about her talk about climate change and the major role of microbes on our planet. We know they have a huge impact on everything we do, from healthcare to agriculture and climate. But there are many gaps in our understanding, and Professor Worden will explain what can be done to address this. Her talk is Climate Change, Integrating the Influence and Trajectories of Microbes, Past, Present, and Future. Alexandra.
Thank you. Well, it's so wonderful to be here live with all of you, and I actually get to not wear a mask, so that's kind of exciting too. What you're looking at here in this watercolor is a painting of an organism called Micromonas. Now, this is about a 2-micron free-living eukaryotic alga, so it's photosynthetic, found all over the marine environments. So just like us, it has all the same complexity in cellular structure, but it's about the size of many bacteria. This is a flagellated cell, and again, it's found from the tropics right up into the polar ecosystems. What this watercolor does not depict is that this cell is surrounded by predators, by viruses, by competitors for resources, maybe by friends and interactions we don't know about yet, and it might have to seek out resources that can be patchy in the oceans. So our goal today is really to understand what happens when a photosynthetic cell like this takes CO2 out of our atmosphere, packages it into organic material. Where does that organic material go, and how might that change in future oceans? So I have kids, and that means I get to read all kinds of books that I wouldn't otherwise have time for. And for those of you who've read The Little Prince, you might remember that he comes to the adults in the room and says, "Do you find my drawing frightening?" And the adults in the room kind of scoff and say, "Well, what is frightening about a hat?" And the Little Prince is very disappointed because indeed this is a really frightening picture. And I feel like as microbial scientists, all of us in this room are really trying to see these interactions that otherwise are quite cryptic, that we can't see or understand that this is indeed a very frightening relationship. When we see two microbial cells together, it takes a lot of work to know whether that's friendly or predatory or pathogenic. And that's what we are all trying to do, I think, in trying to understand microbial worlds, is to make the invisible visible. Okay, so efforts to reveal the unseen really span microbiology. For example, if we go outside my field, we can see some exciting papers. This is 2016, where really targeted efforts to get some of the uncultured microbes, bacteria, into culture revealed extensive sporulation that was probably important in host-to-host transfer. Or papers where they're focusing entirely on uncultured human gut bacteria and seeing how they affect host cholesterol levels. Or a paper like this that's still in the archives, seeing that sulfur chemistry in the human gut actually plays a role in the degradation of xenobiotics. So again, looking to understand what we cannot necessarily see just visually. But what I want to talk to you about today really are the oceans, and we'll talk then about a little bit about oceans today, then we'll move to the past, present, and future. So today, marine algae play a huge role in ocean food chains that, of course, we benefit from as well, and they remove half of the CO2 that is taken up on this planet from the atmosphere. So really important in maintaining this biosphere that we know and love so well. But who and what determines the fate of the carbon of these algae? So if we look here in this food chain, we see these very important algal groups. We're still getting a handle on which ones we need to focus on most, especially as we look towards future changes. We have these whole groups of consumers, these being single-celled eukaryotes. These are the protists. We really don't know which protists feed on which algae, but fundamentally important then for fisheries food chains, again, that we care about so much and that are important for organisms in the ocean. And again, very important is this removal of carbon dioxide from the atmosphere by photosynthetic algae activities of these cells that then sink into the deep ocean and sequester that carbon from the atmosphere. Now, marine algae are responsible for half of global photosynthesis. Half of that CO2 uptake happens in the ocean. And if we look down from space with a satellite, this is what we see. This is an annual cycle being repeated. And what you can see is that annual seasonal greening that we're all so familiar with on land, chlorophyll being detected from space. And then you see these dynamic blooms of algae in the ocean. Now, this isn't one organism. This is many different organisms. And what we don't know is when we see a bloom like this, is that one type of organism blooming, or is it a whole succession as viruses come in and predators come in, or ocean currents change, or nutrient availability changes? And those are all the things we are trying to figure out. Now, I think many of us in this room would consider an oxygenated environment. We think of that as home, right? Home is where there's oxygen for human. And of course, we know that we respire carbon dioxide, and we put lots of it in the atmosphere right now by burning sequestered carbon, basically, from ancient time. But this atmosphere is pretty new to Earth. It's well, maybe halfway into Earth's history, we started having an atmosphere like this. It took organisms figuring out how to use light energy and convert it into chemical energy before we could get to an oxygenated atmosphere. So let's take a look at that. Earth was formed about 4.54 gigaatoms ago. I love that word, but I really only work in the present, but it's just a great word. And what you can see here on the x-axis is billion years of time, 4 billion years of time. Well, we're really stopping at 3. And on the y, you're looking at atmospheric oxygen. We have this event called the Great Oxygenation Event. So what you can see is there's no oxygen in the atmosphere in the first 2 billion years, but then we have this huge event where the atmosphere becomes oxygenated. And there are other little events that are notable too. Now, in terms of history that humans tend to think about, land plants didn't even show up till about 470 million years ago. And so there was no terrestrial place for animals to develop in the same way that they could once there were plants. So I think our lives are intimately and directly connected, of course, with the development of land plant. Now, there's a tremendous microbial backstory here that, of course, is very difficult to make visible because microbes don't make the best fossils. And so when it comes to looking at these, we can sort of name these geological periods. We can say, well, we know that life, we don't know how, but it came to be at some point. And then we had microbes, bacteria and archaea. At some point, anaerobic photosynthesis was developed, and then the cyanobacteria figured out how to do oxygenic photosynthesis, where oxygen was produced as part of the process of fixing CO2. And that is what led to this great oxygenation event. Now, I want to point out that that event that we were probably super happy about actually was highly poisonous. This was a waste product from these cyanobacteria, and it led to the first mass extinction because up until then, organisms were anaerobic, all those microbes. So just think, this massive die-off. And at the same time, there was a reduction of greenhouse gases due to these activities that resulted in climate change, in fact, a cooling of Earth that probably made it a little bit more hospitable. And I just want to point out that this is happening over a period of about 200 million years. And if you look at the changes we're doing right now with CO2, it's a kind of different time scale. We're sort of working on the 100, 150-year time scale right now. And this kind of event that did cause a mass extinction was about 200 million years in time. Now I want to point out, of course, this isn't happening in the sky. A lot of this is happening in the ocean where these cyanobacteria thrived and could replicate. And of course, then it's about this exchange between atmosphere and ocean. So what triggered the evolution of protists, the unicellular eukaryotes that I'd like to talk to you about today, and just like the one in that watercolor? Well, it's exactly this event where life became toxic. The environment became a terrible mess. That sounds familiar, right? That caused some microbes to say, or I don't know if they said it, but they had to figure out another way to stay alive. They had to figure out a way to deal with this toxic environment. And some cells came together and formed the eukaryotic cell. So how did we get from prokaryotes to single-celled eukaryotes and onwards to multicells like us? Well, it's actually still a mystery. What we do know is it happened around the Great Oxygenation event. We do know that symbioses were key in this. So the current idea being that it involved a bacterium and an archaeum, this Asgard archaea, which have a lot of commonalities with eukaryotes that were previously unknown. And somehow, these cells came together, and then we ended up with a unicellular eukaryote that had the structure of a nucleus and a mitochondrion. Now, the classical view is that there was some form of engulfment between these cells. There's a new hypothesis that just came out that it was entanglement. So this Japanese group spent 12 years trying to get an enrichment culture of that uncultured archaean, and it has these bizarre protrusions that they then hypothesized sort of entangle that bacterial cell, and there was a fusion, and it led to this new eukaryotic cell. Now, important in eukaryotic evolution is the fact that there are many other symbiosis events that have happened since. In fact, all the photosynthetic lineages come after one of these cells in a primary event engulfed a cyanobacterium, making the first photosynthetic eukaryote. So what does eukaryotic life look like today? Well, we're over here represented by this whale. We're basically fungi in this schema. So yeast is a great model. What I want to point out is that we have photosynthesis in protists over here in the rhizaria. We have photosynthesis over here in the alveolates. This is where the toxic dinoflagellates that cause toxic blooms that ruin oyster fisheries reside. We have photosynthetic algae over here. This is a diatom known to be very important in fisheries food chains, but we also have an organism like potato blight in this same group. So there's lots going on through evolutionary time that shapes and changes these organisms. We have algae over here that can be detected from space, and then we have the organisms that are the product of that primary endosymbiosis event where a cyanobacterium was engulfed, and that includes the green algae and red algae, and ultimately land plant. And what I'd like you to notice here is that all of these groups are as evolutionarily distant from each other as they are from us. So we think of photosynthesis and as if it's some unifying characteristic. They're really extremely different organisms in terms of their evolution and capabilities. What's sort of the state of affairs in ocean science? Well, we've got diverse marine unicellular eukaryotes. We know they're diverse. We're still discovering new ones. We know that many are morphologically similar, or at least some of them are so small that we can't tell how they're morphologically different. Many are uncultured, and if they're uncultured, clearly their knowledge of their physiology is extremely weak. We're still discovering new photosynthetic lineages, which surprises me, although my lab has done it, but I still don't understand how we can still be figuring things out like that. Their distributions are poorly known, and their interactions with other cell types that dictate their ecology in the environment are unknown. And so today I'm going to be focusing in on an organism over here, the Choanoflagellates, which are very interesting from the perspective of thinking about animal development, and I'll talk to you about that in just a moment, and then on some algae that are over here in the primary endosymbionts. Now, why is our knowledge this weak? Well, as you can see in this same video, looking at algae from space, this is a really expansive environment. It's actually, of course, much more of Earth's surface than land is. So it's pretty hard to cover all of this by ship. The other thing that you're not seeing here is that this is just the very surface of the ocean. But photosynthetic growth extends down 200 meters, whereas most trees are 10 to 45 meters. So there's really big place where we expect to see algae growing, but we can't detect it from space. So there's this tremendous vertical dimension, and that's not just for the sunlit part. It extends down. A typical water column is about 4,000 meters in depth, and then the big trenches are about 9,000 meters in depth. So that, of course, presents some challenges for sampling. And then there are the deep sea sediments, which are, of course, the grave of many organisms, millions and millions of years of settling detrital material, and they're the home of many microbes. They're a reservoir of organic carbon. Okay, another challenge that's really restricted our knowledge is weather and the work environment. So this is a really beautiful flat day out in the North Atlantic where we've moved our equipment onto a boat in order to do our research. Sort of a semi-molecular lab on a rust bucket. And this is actually very flat ocean, but you can see that doesn't mean it's totally flat. And believe me, the ocean is not like this all of the time. And so what happens very often is that people shape their fieldwork by the season. And it's not to target all the seasons, it's because if you only have 3 weeks to collect samples, you really want to go when you can get your equipment in the water, and that only happens if it's not too stormy. Okay, and the fourth challenge, of course, common to everyone in this room, is that often the cells are invisible, or maybe they're in an environment that you can't easily sample, for example, the human gut. And they're uncultured and diverse. So looking through a microscope at seawater that's on a filter, you can see their bacterial cells, you can see eukaryotic algae, you can see viruses, but we don't know which cells those viruses infect. We don't know which cells here are friends or foes. So interactions are difficult to see, and even if you can see them, they're difficult to interpret. Okay, so I think really our goal here is to discover what home means for each microbial taxon. And then maybe you could assemble something and figure out exactly what all the consequences are, the resulting microbiome to ecosystem roles and impacts, once we could define what home is for a particular species. So methods that we use, and I'm sure many people in this room use to view microbial communities, well, there's metagenomics and there's metatranscriptomics. You can certainly want to culture if you can get it, and then you can do physiology and genomics and proteomics and all kinds of things. We do lots of amplicon sequencing, 16S and 18S ribosomal RNA, to try to just assess diversity in the system. Now, the collection methods for these approaches very often are even, for example, fecal material. It's not really capturing it exactly in the habitat that you would most love to explore it in. In our case, we're just filtering down tons of seawater and mashing everything together and then extracting it. So we've also sort of obscured the lifestyle or the home of each microbe as we sample. So what I want to talk to you about today are the methods for discovering specific activities and interactions by individual microbial cells in the ocean. And to do that, what we're going to use is fluorescence-activated cell sorting, or FACS, and we're going to use stable isotope probing. So in the FACS work, what we're doing is we're out at sea and we'll stain maybe the food vacuole of a cell, or its flagellum, or cells that have a food vacuole, and then we'll select them if they also have chlorophyll, if we want to understand which of the photosynthetic cells also eat other particles, and then we'll sort them based on whatever criteria we're interested in. For the stable isotope probing, we're going to grow environmentally relevant prey. So that guy that you saw on that first slide, for example, is one that we grow, and we label it with 13C, so heavy carbon. And then what we're able to do with those realistic prey is incubate them out at sea, and when a predator eats them, it will incorporate that heavy carbon, and then we can run a density gradient where we can pull out the cells that had incorporated that heavy carbon and therefore had to have eaten that cell those prey items versus the rest of the community that did not eat those cells. Okay, so, and here's cell sorting at sea. So we're just going to be running cells single file past a laser. This is showing little tubes because I made it a really long time ago. Of course, now we're doing it in plates. But we're going to separate and capture healthy, wild, photosynthetic, or stained eukaryotes of interest, and then sequence their genomes. And we do this because we need to know about the evolution and ecology of these many algae that we don't have in culture. We can't just study the few lab rats that we have. Okay, and this has been incredibly powerful for looking at protistan evolution and diversity. Our first paper coming out in PNAS in 2010, and our most recent with Tom Richards at Oxford, looking at flagellated cells in the ocean. But today what I want to talk to you about are wild protistan cells that we're going to analyze with their physically co-associated entities that come with them when we do the sort of that eukaryotic cell. Now, I just want to take a little sidestep, a note for early career scientists in the room. So when I applied for my first major research grant in 2004, it was to do exactly that work. That first paper in PNAS, you know, it's not a super shabby journal, exactly to look at the genomes of wild protists at sea. And my reviews came back and said this approach is impossible, it will never work, and that she doesn't have the genomics background to do it, which for those of you in the room who were doing science in 2004, well, I guess the medical field was quite a bit ahead in terms of genomics and microbiology, but there certainly weren't a lot of oceanographers doing it. Somehow I had faith that we could do it. Somehow I got money another way, and a few years later we could show we could do it. The other thing I want to point out is it took a really long time to get to today's data on cell-to-cell interactions, looking at the movement of carbon through the system. And I can tell you that I made up a dreamy website in 2003 when I was a postdoc, hoping I would get a job. And it said I was going to do exactly what I'll be showing you today, and yet I'm showing it to you today, not like 15 years ago. And that's because there was so much homework needed that we needed to do first to make sure we're really looking at the movement of carbon of environmentally relevant algae. We didn't really know which algae were out there and most important, other than the cyanobacteria that could be easily identified. But the eukaryotes, it was still kind of a big mystery. So there was a good 20 years, because we're still doing it, of work to figure out who we wanted to target. First, we needed to identify and characterize key ocean picoeukaryotic algae. Okay, so what we do know now, and I can't spend time talking about it because it's not the topic of the talk, is that these tiny presenophyte green algae are much more important in the ocean than originally thought, and these are the products of a primary endosymbiosis event, and these are intimately connected with the development of land plant. They reflect the progenitor of all land plants, which arose indeed from a green alga. And so they've been really fascinating, not just because of their oceanographic importance, but also because they tell us a lot about the development of plant. Okay, so I'm just going to show you one tiny piece of data about these so to justify why we use them in our later experiments. So I told you that weather and seasonality have skewed our perceptions. So I want to take you up to the North Atlantic phytoplankton bloom, very important for fisheries. This is a region where green algae were not thought to be important. So diatoms that were thought to be important based on data that came from the eastern North Atlantic. And we're working now in the western North Atlantic. And it happens to occur during a really rough time to sample. So this is our ship, and this is a big vessel, just so you know. This would fit about 6 people around it. And yeah, there's a reason that people hadn't sampled in this area very often. But what we learn when we do looking at 16S V1, V2 amplicons, what I want you to see here is just the amount of green. These are these tiny green algae that just were thought not important. They weren't even known to exist up there. And what you can see is that the guys that were thought to be the most important, at least by relative amplicon abundance of the plastid-derived 16S, are not very important. And we can confirm this with flow cytometry experiments. So there's a real importance of strategic targeted sampling. And again, I'm sure that's true for all of our experiments as we think about the human microbiome. So whether it's at the microbiome level or ocean scale, sample collections can drive your conclusions. And the last piece I want to show you about these guys is connected to climate change. What you're looking at here, this is not from my lab, is data on the x-axis representing years. The years on the x-axis, and then you're seeing these tiny prosynophyte plankton increasing in the Arctic, and the diatoms, those larger ones known to connect to fisheries, declining. And what we see here is this very high abundance of one of these small prosynophyte green algae. By the way, this is the chloroplast that was once a cyanobacterium. You can see it, basically, it's just a photosynthesis machine. But what we see is that this increase in Micromonas is correlated with higher water temperatures and reduced salinities due to ice melt. And so what you have to ask yourself is, what are the consequences for carbon export and the food chain? These are very different size cells. We know the diatoms just sink right out of the ocean into the deep sea, removing that carbon. Small cells don't sink. It's like syrup for them. And we really don't know if the fisheries food chains are supported in the same way by these small cells. So we need to know who eats these PQ eukaryotic prosynophyte algae, and we also need to know about the cyanobacteria that are very abundant out there. And so those are exactly the kinds of experiments we're going to do right now. So when we do experiments using FACS or SIP, what we can see is that a group of organisms called Choanoflagellates are out there eating. And these are unicellular predatory protists. We see them when we FACS sort cells with active food vacuoles, so not sort of amplicon sequencing of everything that's there, but just targeting the cells that we're eating in that moment. And when we use SIP, we see Prochlorococcus and these green algae being incorporated into the DNA. So two different methods for doing this. And I want to just point out here that 18S relative amplicon abundances really don't shine a light in these guys. You just would think that totally other predators mattered in this system, and that could be a copy number thing, it could be other biases, or it could be they're not very active at that time. So it's really important that we really try to get to active cells. Now, what are choanoflagellates? So they're predators of bacteria. They've been known in the oceans for a long time, but not thought to be very important. They're the closest living unicellular relatives of animals. And they are so neat. So this is a single-celled choanoflagellate that we've been sorting, but they can form a truly multicellular state. And for that reason, they're heavily studied to try to understand the development of animals. How did we get to a multicellular state? But I want to talk to you today about how our single-cell genomics has exposed that these unicellular marine choanoflagellates have a microbiome. So if we go look, after we've single-cell sorted all of these, at the inner world, so what we're using is, again, the FACS sorting live protists that have their food vacuole stained, whole genome amplification, sequencing, and genome assembly. What we see in this particular experiment is Bicosta minor. It's an uncultured choanoflagellate. It was in 99% of the individual wells that we sorted to. We see a giant virus in some of the wells. That was really exciting. I don't get to talk to you about it today. It's all published. I thought I should share newer things. But if you're interested in viruses that have genomes the size of a bacterium, check out that work. But what we see is this divergent gamma proteobacterium. It's present in 13% of the choanoflagellate cell sorts. But it's very rare in seawater. When we just filter down the seawater, it's about 1 in 40,000 ribosomal RNA genes. So it's very unlikely to be a prey item or prey bacterium because this choanoflagellate would have a hard time encountering it at those frequencies. But of course, we wanted to know more. So when you look at it from a phylogenomic perspective, what you see is the cocciolales, and here you have the legionales. And you can see that there's this group, and here are the two guys. We sequenced 2 different genomes from these choanoflagellates, and they're falling in this group of all marine and all uncultured bacteria. So we don't want to do guilt by association, but it is related to animal pathogens, so that was something we wanted to follow up on. Now, something that was very important to us is to know that we would have a complete genome, because if you look at pathogens, you might see reduction in genome, but then you want to be able to attribute it to true reduction and not just patchy data. So we finished these genomes. These are closed genomes. You can see the origin of replication, and they're complete. And you can see they're quite small. What you're looking at out here it's metatranscriptome data mapped onto those genomes showing that all of these genes are expressed. Now, if we compare these genomes to, for example, marine isolates, we see that they're very small. If we compare them to metagenome assembled genomes from the environment, we see that they're also small. They're more similar to some of the single amplified genomes, but those tend to be rather incomplete. And what we think is we have this beautiful complete genome because we weren't sequencing one bacterium. We were sequencing one eukaryote that had more than one clonal bacterium in it. Okay, now one of the really fun things about this guy is it has a type 4 secretion system, but it's a specific subtype. It's the same secretion system that we see in the animal pathogen Coxiella burnetii, and it's missing the relaxase that would allow it to be used for conjugation. So this secretion system is being used to pass effector proteins in all likelihood to that choanoflagellate in which we saw the cell. So you can see the synteny here with Coxiella, and again, you're looking at heat-mapped transcript expression in the environment. Now, when we look at a phylogenetic perspective at 3 of these genes that are critical in this system, you can see that a number of the other uncultured marine group members in that group that I showed you also have this secretion system. And you can see Legionella and Coxiella, again, not our most friendly characters. Okay, but this secretion system, when we look at secretion systems across all marine bacteria, it's really rare. In fact, it's super, super, super rare. And so we have to ask ourselves, is that because it's rare? Or is that because really the things we've been sequencing come from this big mishmash system, and if you're a rare cell, you're never going to get your genome assembled. If you're a rare cell living in a protistan host, probably you got thrown away in the data. Not that there's a lot of people doing this kind of single-cell work yet for the protists. But what I want to show you now with choano, this divergent gamma proteobacterium, what you're looking at here are pathways down the Y, and a bunch of known characters like E. coli over here, and some intracellular obligate symbionts here. Now, someone like E. coli is very enriched, as you're seeing with the color here in these pathways. It might even have sometimes more than one copy needed for a particular pathway. And then we have someone like Legionella and Coxiella, all very enriched in these pathways. Prochlorococcus, the most abundant free-living photosynthetic organism on the planet, that's cyanobacterium we've been working with, also complete in these pathways. And what you're looking at here in purple are other members of that uncultured group, and in stars, the choano that we found with the choanoflagellates. So when we look here, you can see the obligate intracellular guys really missing a lot in these pathways. We can see choano also missing a lot in several pathways. And what I want to point out is that this bacterium doesn't have a single amino acid synthase, so it's completely host dependent for that. Its glycolysis pathway is broken, it's missing multiple genes, and we know this is a complete genome. And it's missing fatty acid synthases, it's missing LPS synthesis. And of course, what we know from pathogens is that often you can see reduction in LPS as a way of evading host immune responses. What we do see is that the choanoflagellate is able to take ATP from the host. So choano is a pathogen or an energy parasite. We haven't yet figured out if there's anything this cell is giving the choanoflagellate host. Nothing has become apparent. But what we can say is it's using resource from the host. And I think we have to ask ourselves, when we have this huge clade of uncultured marine bacteria, is that really just because we're never targeting the right environment? Is it because there was never a host in those culturing efforts? And so it was completely, they've been sort of homeless of unknown ecological niche until now. So what we have is the Choanoflagellate. We have this divergent obligate symbiont, the gamma proteobacterium. What we think is going on and why we have such beautiful genomes is that it's replicating inside the cell, parasite or pathogen. And the key thing to think about here is that these Choanoflagellates eat bacteria for a living. So in every food web model, that's how the picture is drawn. The carbon moves from the bacteria into the Choanoflagellate and then up to the fish. But what we're seeing here is that the bacteria is kind of using the resource of the Choanoflagellate. We've flipped the canonical flow of carbon in this case. Now, I want to say that we've also found a divergent symbiotic alpha proteobacterium in these cells that I won't have time to talk to you about and that we're still following up on. Okay, so what we've learned now as we try to understand controls and interactions and fate is that there are these two big areas. I didn't talk to you about the giant viruses, but these symbionts or pathogens that are intimately involved with the Choanoflagellates that belong in this heterotrophic protist box. And what I want to end the talk on is talk to you about active degraders in the deep sea. What happens when an algal cell moves into the deep sea? How is it degraded, or is it degraded? And this has been a big team effort that we've been pushing on for quite a few years, and this is still fairly preliminary data. So what we're going to be looking at is export and carbon processing deep in the sea. So an algal cell that sinks to the deep sea. And we're asking which members of the sediment bacterial community are active, which can actually respond to an algal cell that shows up there. And this is an important question because the deep ocean is 2 to 4 degrees C. It's like a refrigerator. So any DNA that falls down there doesn't necessarily get chewed up and used immediately, so you don't really know which part of the community is active. So the devices we're going to be using were something that came out of an interaction with Tom Richards at Oxford. And he said, Alex, look, every time you sample the deep sea and bring it up and do metatranscriptomes, people say, oh, well, it's all garbage because you just took it up 4,000 atmospheres and you took it from 2 degrees to 20 degrees, and why would we believe any of that transcript data? So he said, I want a way that I can preserve the transcripts in situ and bring them back basically fixed, and then process the sample. So he wanted to avoid the massive pressure and temperature changes and the perturbation of geochemistry that happens when you take things up from the deep sea. It's quite a long transit time to bring it up too. And so luckily I just had my kids. I was very foggy all the time. I really couldn't write papers at all, but I could think about cool instruments and how we could use a remotely operated vehicle, basically a robotic submarine, to deploy them. And so what we came up with were these injector cores that we can push into the sediments, and you'll see needles that are going to put this fixative solution, RNA later or Lifeguard or one of those types of solutions, so that we can bring the transcripts up fixed in real time. And so what you're looking at here now are, and that was we published that with Tom, or he published it with us in 2020. And now we're going to move to looking at C13 labeled detrital algal material of these environmentally relevant algae. And so what you're looking at here actually are whale vertebrae. This is an area where a whale had fallen and decayed about 15 years ago now. And you're looking at us pushing these injector solution, and then we can leave it for 2 days to 4 months when we're going to bring it back to look at how bacterial activities changed. Okay. So what's really neat about this is the first thing we can look at is whether the organisms use that material by looking for respiration. So we're going to take that, we have labeled organic material, and we're going to now look for CO2, dissolved inorganic carbon that has the 13C signal in it. And when you look then into this push core, here you're seeing the delta DIC. That says these cells have taken up this material in 2 days' time at 2 degrees C and used it and respired it. And this is a second tracer. This is heavy water that we add just to see where our solution went. So you can see kind of a peak solution here. And now we'll go ahead and sequence and do stable isotope probing. And what you can see is that we have a number of novel candidate phyla that have come up in the stable isotope probing. And it's not, in fact, these very abundant Bacteroidetes that are responding to this new, fresh algal material. Now, so what we're seeing is that rare players kind of popped out of the system, that rare biosphere, and incorporated that 13C, and again in just 2 days, which is kind of completely unexpected for the deep sea. It's thought that things go very slowly down there. Okay, and I apologize that we don't have all the replicates on this slide, but what you're looking at now is just forget the SIP. We're just going to look at relative amplicon abundance. This is Z-score. And so what you can see is that out of 55,000 different 99% OTUs, close to ASV but not quite the same thing, about 12,000 or 1,300 responded. At T0, T2 days and 4 months. And what you can see is this early responder group that in 2 days has increased in relative abundance. We have a late responder group that increases in relative abundance at 4 months. And you can see that many of these taxa are the same as what we saw in the SIP data. This is just showing you particular ASVs and in the groups that match with the SIP data. And then we have these more mixed groups. And it's sloppy because these sediment cores are not homogeneous by any means. They're mud, sediment, crazy stuff. Okay, but what's really neat here is that we have this Begiotoa, not very good at saying that, which is a gamma proteobacterium that Ukrainian biologist Winogradsky discovered about 150 years ago because it's gigantic. It's very important in oxidation of hydrogen sulfide. And this guy, we don't actually see in the SIP data, but we do see it in metatranscriptomics, and we do see it here in this analysis just by looking at changes in relative amplicon abundance. And so metatranscriptomics show that we have increased expression of glycolysis and peptidoglycan, all the things you think of as being key to microbial growth. We see this triggering of sulfide oxidizers and overall sulfur cycling. And at the same time, we see that the hydrogen sulfide that was quite high in these sediment columns is actually completely depleted over time. And again, this especially massive response from these Begiotoa that you can actually see with your naked eye because they're so large. And I want to say this is likely a secondary response because, again, they didn't incorporate the actual label, or at least we don't think they did. So we have this unexpectedly rapid response of specific, rare, and novel candidate phyla about which we knew nothing before these incubation experiments in the deep sea, in situ. We have this response to fresh organic matter influences on the geochemistry within days. So things are happening down there a lot faster than we thought. So I just want to end talking just for a moment about the future. And I think all of us in this room have a tremendous sense of urgency about what's going on with our planet. And that biosphere we call home, that oxygenated place that isn't super hot, doesn't have so many greenhouse gases. And what I want to point out to you, I've shown you that algae live over that whole water column. Actually, in that North Atlantic bloom experiment, we were looking at different sites and over the different depths. And what I want to show you is that from 0 to 200 meters, we have algae growing. That's what this represents. And in this case, they're trading off access to light that they need in order to get these nutrients that come up from below. And as oceans warm, the thermal stratification becomes much stronger and shallower, and those nutrients can't penetrate from below. So you have this sort of huge depletion of nutrient in the surface ocean where the phytoplankton need it. And this is thought that it should select for smaller algal cells, but at some point, everyone needs nutrients, right? And I think the important thing to think about as we sort of apply ideas of selection and who should win is that this isn't the only change that's happening. When you change the water column, the whole algal growing region, you're changing their exposure to light, to UV light, the amount of time they're exposed to that light. You're changing the temperature, which might select for different characters. You're changing the acidity of the water, which makes these green algae lose their flagella. So whatever role they play, whether it's in sex or in avoidance of predators or in trying to meet nutrients, losing your flagella, if you've had it in the evolutionary time, doesn't sound like a great idea. And we know that it's going to shift communities and, of course, reshape interactions. So how will changes shift photosynthetic communities, reroute algal organic carbon that we've just been exploring, we're only just learning about, and algal export and overall food webs? And we've seen today all of these different things, again, totally new knowledge that we could never had in any model yet. And then, of course, I haven't been able to talk to you about the viruses over here today. So I just want to finish by saying that I think the approach here is we need to take this integrated approach, in this case, expeditions. In another case, I don't know, I know it's really hard to sample human and they have to be sick and having surgery, and that might skew your sampling too. And then we have to pair that with model cell systems biology, because it's critically important that as we get into modeling, we have functional information and realistic constraints. It's really easy to make models to explain anything, but without realistic constraints, the models are probably not telling us the right thing. And so, again, working with the wild, mechanistic understanding of growth, and then I think the weakest here is critical for scaling and for developing hypotheses and then testing them, whether it's back in the model cell system or in the field, is the modeling. And I want to say you could replace this with gut microbiome, and you probably have the same circle of things that you're going to need. I mean, not quite the same. You don't need a ship, luckily. And you don't have to work in a rust bucket. But anyway, I want to thank my group. They're totally wonderful, and whenever administration becomes a nightmare, well, at least you have your group to make happy, and they make you happy too. I want to thank my funding sources and various collaborators, and I really wanted to thank all of you, since I know you might be hungry or thirsty, and you've shared your time with me. So thank you.
I think you'll all agree that was an absolutely fascinating talk. I'd like to thank Alexandra for giving that talk. It's absolutely inspirational. Thank you again.
Thank you.